Good morning from Europe and good evening for the Asians and so on. Um, it's a great pleasure to be here. I'm sorry about this kind of ugly title. What I usually call this presentation is War and Peace in a Microfluidic Droplet, but I thought maybe uh, that's not very easy to understand. So what I will talk to you about today is how can we actually learn something about biology by leveraging things that you can only do with microfluidics, basically. So the way to think about this for me is that if you go to a biology lab, typically you have very different types of machines. A lot of the measurements that people make are on the population scale. So at this scale, it's very easy. You put cells in a tube, maybe you put an antibiotic and you ask, did the cells grow or not grow? And you get a quick and easy answer, which is in many cases the relevant answer because, for example, in the case of infection, what you want to know is do the bacteria grow or do they not grow? And on another scale, people have been developing many techniques to work on the single cell scale. And on the single cell scale, traditionally, this has been done with microscopy. So you look at the cells and you ask, what are they doing? And then many single cell technologies have been developed to kind of increase the number of things we can do on the scale of individual cells. So single cell biology for a long time was a very hot topic. And people thought that, oh, okay, so if I know what every one of the cells is doing, I can just kind of combine them together and get the population scale. Except that it turns out actually linking these two extreme scales is very difficult for many reasons. One of the reasons is that a tissue is not just a bunch of cells. The cells are interacting with each other. So if you have n cells, it's not enough to know what each one of the cells individually is doing. You have to know what the interactions are, and this becomes an extremely complicated problem to solve. You have to account for interactions that could be mechanical if the cells are touching, it could be chemical if they're secreting something, they could be actually transmitting electrical signals or other types of biological interactions. And in particular, when you have when you want to build from single cell to many cells, you have to take into account space and time. So things that happen locally at one point in time do not necessarily affect everybody at the same time. The signals have to transfer, and so this becomes also a complicated problem. And so one way to start bridging these two scales is to work at an intermediate scale. And I don't know what to call this scale. I usually call it the collective scale. So at this collective scale, you imagine you work with a few cells or a few hundred cells or a few thousand cells. For these numbers, you continue to be sensitive to what each one of the cells is doing. You continue to be sensitive to the heterogeneity between cells, but you start observing the interactions between the different cells. And so you can build not only the individual cell behavior, but also the interactions between them. But working at this scale is challenging for many reasons, and maybe two particular challenges are that there are not so many technologies that allow you to work at this intermediate scale. So fax machines and microscopes are one thing, and PCR machines are another. But then between the two, it's at least until recently there was a big gap, and because of this big gap, then there's also not so much conceptual framework to build this interactions. And so what we wanted to do in my lab a few years ago was to build technologies that allowed us to work at this intermediate scale. And then also to ask, well, what can we measure at this intermediate scale to help us bridge the gap between individual cells and organisms? And so we're definitely not the only people doing this. There's many others. For example, if we think about microbiology, so bacteria and how they organize together, if you have individual bacterial cells, you can see them on the microscope and you can kind of observe them. And then when they come together, one of the multicellular organisms for bacteria is biofilms, where things are extremely complicated. There's many chemical, biological gradients, and so on. And so making the link between what an individual bacterial cell is doing and the biofilm is doing is extremely difficult. And but there have been some really interesting work that has been done. This is one paper that I think is really beautiful, where they grew a bacterial monolayer inside of a microfluidic chip. And by observing calcium signaling between the cells, they could observe electrical signals being transmitted by bacteria, by E. coli, in a way that reminds us of kind of neuronal transport. So electrical signals between bacteria were discovered in this way. If we go to the mammalian cell world, again, this distance is extremely large between individual cells and organisms. And between the two is a very, very important and hot topic of microphysiological systems like organoids and spheroids and everything that is being built around those. And so there have been very important technological and fundamental developments to bridge these scales. But then you could ask yourself, what do actually people learn from these assays? And from my point of view as a physicist by training, I find that it's a little bit disappointing in some ways because organoids are mostly treated in bulk as if they were individual cells. And organs on a chip, usually people get one measurement from a chip. And by getting one measurement of the chip, they are treating them a little bit like animal experiments where you spend a lot of effort and time to make an experiment and then you flush the contents and you measure it on an ELISAs or you measure some kind of sequence. So the idea is, can you learn more from these experiments by developing different analysis and manipulation tools? And maybe I think this is my last introductory slide. If you want to do this, you have to combine together many different aspects. The first aspect is the microfluidic device. It's true, you have to develop your lab on a chip or organ on a chip device, and you have to develop protocols on how you're going to manipulate the biological samples. You have to add to this. So this would be maybe the ground floor. And then the first floor would be what actual biological contents do you put in there? And there we are seeing in the community more and more people working with stem cells, with patient cells, primary patient cells, and organizing co-cultures into these devices. And so this is something that is actually adding a huge amount of value to these platforms. But then, from my point of view, there's also a last part, which is what kind of data do you get and how do you analyze these data? And this is where I think that there's a lot of work that still could be done. But in the end, at the end of the story, what I think you have to remember is that it's not enough to just have a device. It's not enough to just have the cells, and it's not enough to have the analysis. All of these have to work together in order to really bring transformative new knowledge. Okay, so the challenge that I'm going to try to address today is how can we relate these different scales by performing kind of deeper physical and biological measurements and combining those with mathematical modeling, which are necessary if you want to actually extract information from all of these data. So I will show you two examples. I will I will not go in a lot of detail, but I will show you two ways in which we are working along these lines. The first one is to use stationary droplets to study the response of E. coli to antibiotics. And so this is a long project, but most of the results I will show you today were produced by Lena Lekelek, Andrei Aristov, and Eric Micrans. And this data are in an bioarchive preprint that you can download. So before I really go into what we do in the lab, I would like to remind you that every time you think you're doing something really original, it turns out somebody else did it 50 years ago or 70 years ago. So I was told recently, a few years ago, that we're not the first people to do droplet microfluidics. In 1950, at Institut Pasteur, Andrei Lvov, who was a Nobel Prize winner from Institut Pasteur, wanted to study interactions between bacterial cells, in particular, how do phages that infect individual bacteria transmit to other bacterial cells. And for this, they had to find conditions that were very pure. So by reducing the volume, you reduce the chances of having unwanted cells in there. And they wanted to be able to add and remove things from these samples. And apparently, so I don't know this person, but apparently there was a really brilliant engineer called Monsieur Fonbrun, who developed a micromanipulator where they could deposit individual aqueous droplets on the bottom of a microscope slide. And each one of these droplets, if I made my calculations correctly, was 0.01 nanoliter. So this is very, very small droplets. And then they could put this on the microscope and they could observe them. And in this way, they were able to discover how lysogeny works, and they won the Nobel Prize. Well, Lvov won the Nobel Prize, but along with his collaborators. So the more 21st century version of this is a microfluidic chip that looks like this. This is the chip. This is a 10-cent coin, so it's only maybe 5 millimeters. Well, 1 centimeter. um, and in this chip, what you have is you have two inlets that are shown here, and then you have a region where we have very thin chip, but on top of the chip, we have little pockets. We call these anchors in the lab. And so in this chip, we have 500 of them. And if each of them has a volume of about 2 nanoliters. The way we do these experiments is that we start by filling the complete chip with oil. And then once it's full of oil, we fill it with an aqueous solution that contains the bacterial suspension. And then we bring the oil again, and then by bringing the oil again, we break the little droplets, aqueous droplets, inside each one of these little pockets. So a movie that shows you this that was done by Gabriel Amsalem, these are the little anchors, the little squares. You can see here the interface between the water and the oil. And so this is the second step. So we have filled it with oil, we have filled it with the aqueous suspension. And now when we push the oil again, and this is time in seconds, so you can see that it happens relatively quickly. What you find is that each one of the anchors breaks one droplet inside it. Each one, not exactly each one. So sometimes you have little artifacts like this where it didn't break properly. But in general, we have one droplet per anchor. Some of them are a little bit bigger, some of them are a little bit smaller, but it's a very easy thing to do. And you can do this by hand. You can fill the syringes by, push the syringes by hand, and it still works. It's very, very robust. And so, okay, this is what I just said. The nice thing is that you can then put this on a microscope. And here what I'm going to show you, so these are 3x3 droplets containing fluorescent E. coli cells. And this is on a spinning disk confocal microscope. So we're doing Z stacks and we're doing Z projections. And what you see is that you do time-lapse movie and you can observe the growth starting from 1 or 2 or 3 cells of little colonies inside each one of the droplets. And you can see that even though these are very homogeneous cells, isogenic from the same culture, you have different morphologies. You have clumps in some droplets, and other droplets you have only planktonic cells. And then in many droplets, you have combination of clumps and planktonic cells. Now, if I put some antibiotic just a little bit below the concentration to kill the cells, what you find is that the cells still grow. They grow a little bit slower, but they grow in a very different shape. Because this is cycloxacin, it's well known to produce filaments, but then the filaments can also lead to planktons, and then the cells actually, you have a combination of different morphologies in the droplet, and these morphologies are affected by the presence of the antibiotic. So the reason question that we've asked is how does the cell morphology depend on the antibiotic presence? Sorry, it's winter. So the way we treated this was to build a holder that we could do 6 experiments at the same time. So each one of the experiments that we now do contains 6 conditions. Each chip contains 1 condition. So we tried 5 different antibiotics, and in total we had 168 antibiotic concentrations and conditions. This generated images of 80,000 individual droplets, and so each experiment was 60 gigabytes. And so by doing this, we generated lots of data. And then by looking at the data individually, we could by hand detect that there were maybe 5 big categories of morphologies. One of them is to ask, is the droplet positive or negative? Because for large enough antibiotic concentrations, the cells just don't grow, and so the droplet is negative. But if the droplet is positive, then we could identify 4 different morphologies: planktonic, ones with filaments, ones with rods, which are very short filaments, and ones with clumped cells. And so depending on the droplet, you could see very different morphologies. And these morphologies, we think, are really a signature of the mode of action of the different antibiotics. And so the challenge now is, of course, you want to use AI to analyze these 80,000 droplets, but the challenge is how do you actually interact with 80,000 droplets with tens or hundreds of gigabytes of data? And so I'm going to do a really risky thing. I'm going to do a live demonstration. So Andrei Aristov, our engineer, he developed a platform to actually interact with these data. And this is the risky part. I'm going to interact with the platform. So the platform is online now. And you can see we have the 5 antibiotics. So let's just pick gentamicin, for example. And these are the data we have for gentamicin. And if I pick, I don't know, this one, what you see is the complete chip. And you see individual droplets on this chip. This is day 2. I could also switch to day 1. So this is the initial state of the chip, and this is the second day. So on the second day, what you see is that the droplets, a lot of them contain cells. Some of them are shrunk, so we can label them. So with this platform, I can label individual droplets. So I can exclude, for example, droplets that shrunk like this. I can also ask the software to show me the first day and the second day for the same droplet. And in this way, do kind of a quality control on each one of the droplets in each one of the chips. I could also, so this is one way to do quality control on the data. There's another thing that you need to do is to label what is inside each one of the droplets. And I can do this with context-unaware labeling. And what this does is it takes all of the data from all of the experiments and selects randomly certain droplets. And then I can go to these droplets. So this one is positive and contains planktonic cells. This one I would say is positive and contains filaments. And I can this way train very, very quickly many droplets in order to train a neural network to identify the contents of the drops. So this is software. So again, the paper is on bioRxiv, and you can download the software and the data and play around with them. And by doing this, so you train a neural network, and then you go back to all of your droplets, and then you can ask, as a function of concentration of antibiotic, how many droplets are positive, how many droplets contain planktonic cells, and so on and so forth. And so you can, for each one of the experiments, detect how many are in which condition. And this is ugly data. You don't really know what to do with them. But I'm not going to show you a real biological conclusion, but what I will show you is that by combining together these different measurements, what you can find is that for each one of the antibiotics, as you go from low concentration to high concentration, you go through morphology space in a complicated way. And this is really interesting because what this is telling us is that you can calculate, you can measure multidimensional morphologies for each one of the antibiotics at different concentrations. And this is a way to identify how the antibiotic interacts with the cells. And so if you have an molecule that you think is going to be useful as an antibiotic, you can run tests like this and see how does the morphology change, and you can then relate it to what different known antibiotics are doing and kind of get a mechanism of action this way. And so this is very much engineering work. We've standardized the microfluidics, we've standardized the analysis, and now we can run many different molecules and try to understand what they are doing. And we're also applying very similar analysis to yeast cells and to different types of microorganisms. And so we think that this is a platform on which we can build for doing really new types of biology. Okay, so this was the first part of the presentation. The second part, I'm going to switch to a completely different subject, which is the subject of cancer, in particular immune cancer interactions. And this is, again, a long project that has been going on for several years. But the recent work that I will show you is done by Valentin Bonnet, Emmanuel Angelikadakis, and Roxane Cronio. Okay, so I think maybe everybody is familiar somewhat with adoptive cell therapy. The idea is that for a cancer patient, in order to treat the cancer, one way to do this is to take some of the cells, for example, the T cells from the blood, to retrain the T cells by reprogramming them to recognize the cancer, and then re-injecting the cells into the same patient. So this is called adoptive cell therapy. There are many ways to do this, but this is kind of the general idea. Um, so this is maybe a revolutionary way to treat cancer. Um, okay, I have a better slide. Sometimes it leads to spectacular improvements for the patient, and sometimes, unfortunately, it doesn't work. And many times when it doesn't work, it's not very clear why the treatment is not working. From a very physical point of view, you can think that in order for the T cells to actually destroy the tumor, they have to go through a different sequence of steps. And here I'm only talking about once they are around the tumor and not talking about systemic viewpoint. But so if they want to destroy the tumor, they first have to migrate and find the cancer cells. They have to recognize them biologically by making the right connections on the surface. And then they have to accumulate on the tumor, and then the killing happens. And so identifying which one of these steps is the rate limiting step is a very difficult thing to do in vivo, and it's not really possible to do on two-dimensional layers that are usually used. So can we use three-dimensional cell culture to identify which one is the rate limiting step? Also, we know that T cells have a very heterogeneous function in the body. Can we identify the different roles that are played by different types of T cells? And can we identify collective behaviors? And so this is what the types of questions that we have been asking in my lab on adoptive cell therapy. The way we do this is again with a microfluidic chip that has three parts. One part is for droplet production, a second part for droplet distribution, and then the real experiment happens in this part, which is the trapping and observation region of the chip. So the way that the experiment works is like this. This is in real time. We generate droplets, and the droplets contain a bunch of cells inside them. So as the droplets are being transported, you see that they're very nicely monodispersed. They contain cells inside them, and now we are in this region of the microfluidic device. And as they reach the trapping region, you can see these little hexagons. These are the traps that are going to actually be used to keep the droplets stationary. So when the droplets reach the traps, they go inside them, and they stay there for the rest of the experiment. The reason why they are trapped is because in between the traps, the droplets are squeezed. And so when they see a region where they can actually reduce their surface area, they just go inside and they stay there to reduce their surface energy. So this was in real time. So in 50 seconds, we started filling maybe 3 or 4 of these rows. So in about 5 or 10 minutes, we can fill the complete chip. Once the chip is filled, we stop the flow and we let the cells sediment to the bottom. And then if we look by time-lapse imaging, we see the cells sedimenting and starting to join together, and they form, after a few hours, a nice sphere that's a spheroid that contains the cells that were in the droplet initially. And you can see that this is a sphere because since this is an aqueous droplet in oil, you can flow the oil around this and you can make it turn, and you see it's a really nice sphere of cells inside each one of the droplets. So these are really old movies. They're maybe 10 years old. So what we're doing now is we can generate now an array of droplets. Each droplet contains, in general, one spheroid. And we can put this on a microscope slide. And so this is really well adapted for microscopy. And so by doing basically scanning on a confocal microscope, we can generate single cell data, but within the context of each one of the spheres. And then by using some of the bioinformatics that we've developed, we can relate each one of the cells with its neighbors, and each one is related to its complete spheroid. And so by this way, we generate what we call multiscale cytometry. Cytometry means we measure things on individual cells, and it's multiscale because we can relate things across single cell to the organoid scale. Right. So a little bit slow today, so let me speed up a little bit. So, and then we can also use little side pockets of the droplets to bring a second droplet. And then by bringing two droplets together, we can merge them together and have different conditions in different anchors. For example, we can use this to screen two molecules at the same time. So these were experiments that were done with cancer spheroids and screening for etoposide and cisplatin. And we can see a synergetic effect or not between the two molecules that are being injected into an individual droplet. So all of this is now published. So I will go past. What we're going to be interested in now is to inject not a molecule, but other cells. And so we can make a cancer spheroid, but now it's not in a liquid droplet, it's in a matrigel droplet. And then the second day, once we've made the spheroid, we bring a second droplet that contains T cells. And by fusing the droplets together, we inject the T cells into the first droplet. And this is what it looks like. This is the cancer spheroid, and the green dots are the T cells. And the T cells, once they enter into the second droplet, they start exploring space. This is what they do. And what you see is that they come in and out of the focal plane because this is in 3 dimensions. And okay, in the first 3 hours, it's really nice to see the T cells moving, but nothing really major happens. But then if we wait a little bit longer, this is the same experiment, just played a little bit faster. What you see is that the T cells find the cancer spheroid, they accumulate on it, and they start killing it. And you can observe all of these effects on the scale of individual cells, and you can observe them on the scale of the spheroid being destroyed as well. And so we can identify from movies like this, the first phase, which is where the cells are exploring the space, the second phase where they accumulate on the spheroid, and the killing. And we can then measure quantitatively each one of these events happening at the individual cell level. But since this is one droplet out of many, a way to think about this is a little bit like a Monte Carlo experiment. It's not a Monte Carlo simulation, it's a Monte Carlo experiment. So we have many times the same experiment with slightly different initial conditions, slightly different number of cells, and we can then build a probabilistic theory to actually describe what is happening. And by doing so this is a long study that I'm just showing you a very short result from it. We can identify long-range interactions that are happening through cells communicating with each other with secreted cytokines, and short-range interactions that are happening by cells being close together on the spheroid. And we get probabilistic viewpoint of how likely is it that the cells will kill the cancer cells. And the probabilistic model fits very nicely with the experimental data. Okay, so all of this is on mouse cells. And the last work that I'm going to show you, which is unpublished still, but we're working on the paper right now, is how does all of this technology translate when we want to use human cells, so patient cells. So what we are doing to understand human cancer cell and T cell interactions is we start with a patient-derived organoid, which we obtain from our collaborators in the hospital. And then these organoids, we transfect them to express CD19, which is a surface molecule on the cancer cells. Facing this, we get CD4 or CD8 naive T cells from a healthy donor. And then the first experiments I will show you is that we can add a T cell engager, which is a molecule that binds to the tumor antigen and binds to the T cell, and so that the T cell recognizes the tumor cell. So this is just a molecule we put in solution with the naive T cells. And we play the same game. We take the cells, we put them in droplets, and then we inject the T cells inside of the droplet containing the cancer cells. And in the presence of the T cell engager, we see that the T cells, it's a little bit less easy to see on this video, but you see that the T cells are migrating, and as soon as they reach the cancer organoid, they bind to it. And we see that we can titer basically the effect of the T cell engager. So we see a dose response as a function of the concentration of the T cell engager, and also a dose response as a function of the number of T cells that are able to reach the organoid. And so in this way, we are doing two separate dose responses in the same experiment that are dosing the T cell, the number of T cells, and the quantity of the molecule that we are placing. And so if you have a new molecule that you want to evaluate, this is a nice way to evaluate the efficacy of this new molecule. And then finally, we can play another game. Well, it's not a game really. I'm sorry to say that. So we can change our paradigm and use CAR T cells. So these T cells that were engineered to actually recognize CD19 directly. And by doing the same type of experiments, we can find that by placing the CD19 transfected cancer cells with T cells that recognize CD19, they are able to migrate in the matrigel. And once they reach the spheroid, their velocity slows down very significantly. What this means is that when the T cells reach the spheroid, they stop moving and they start actually interacting with the cancer cells. And then, so we can do all of the controls that show us that the interactions are very specific. You do have T cells killing cancer cells when it's not specific because T cells are cytotoxic. But when you put the CD19 molecule, you get a much stronger response. And then finally, the last thing I will show you is that can we actually use this to understand better the interactions between the T cells? And here what we did was we separated the CD4 T cells, which are known to be basically helping regulate the action of the CD8 cells, from the CD8 cells, which are the ones that are typically known as being the ones that are directly cytotoxic, the ones that directly kill the cancer cells. So we separate them, and we can do experiments with only CD4 cells, only CD8 cells, or a combination of the two. And by doing our statistical measurements, what we observe is that only CD4 cells do lead to some killing. CD8 cells on their own lead to better killing, and CD4 plus CD8 cells lead to even better killing. However, these bar plots are hiding actually a different type dimension. And we can explore this other dimension by asking how many T cells were observed on the cancer organoid at the end of the experiment. And what we see is that, so these data are a little bit difficult to understand. I'll try to go through them a little bit in detail. If at the end of the experiment we see more CD8 T cells, we see that the viability of the organoids is reduced. So the more T cells you have, the more killing they produce. However, if we do the CD8 T cells on their own or the CD8 T cells with CD4 T cells, and we look at the viability, it's independent of the condition as long as we're looking at the number of CD8 T cells. However, if we do the same thing with the CD4 number of CD4 T cells, we see that the more CD4 T cells we have, yes, the more dead is the organoid. But in the condition where we have CD4 and CD8, the effect is much, much stronger. So the way to understand this is that it's the CD8 cells that are doing the killing, and the CD4 cells are there to help them accumulate. And the helper side that tells you that the CD4 cells are helping them accumulate is shown here. What we see is that the CD4 cells, when they're on their own, accumulate at a certain rate. So at 0 hours, 3 hours, 6 hours, and 9 hours, the number goes up. But it goes up basically the same way whether we have the CD8 cells or not. On the other hand, the number of CD8 cells, if we don't have the CD4 cells, it goes up regularly, but within the presence of the CD4 cells, they go up faster. So these are the state of the data we have right now. It's still preliminary data, but I think it's very promising. What we are seeing is a different role of CD4 cells and CD8 cells inside the same droplet. Okay, long presentation. So what did we learn so far? What we learned is that from the technological point of view, we can obtain spatiotemporal information about single cells by putting them on a microscope slide inside of droplets. By analyzing these data, we can break down the interactions into different phases. And then really what's really useful behind all of this is the probabilistic modeling that allows us to actually distinguish between different hypotheses that allow us to really understand what's happening biologically. From the applications point of view, we can do these experiments with a small number of cells. So we could imagine using this for personalized medicine by taking cells from a biopsy And then we can evaluate the efficacy of molecule-cell combinations. This is an interesting way to screen many conditions in a single experiment. And then what I didn't show you is that we can also measure what's being secreted by the cells, so we can extend the number of things we can measure in an individual droplet. So, okay, I started my presentation with a little bit of philosophy. Let me end it with a little bit of philosophy as well. What I think was key to be able to advance in these projects, the first thing was the microfluidic device. And then we had to basically increase the complexity both on the cancer compartment by going from mouse or spheroids to human organoids, and also from the T cell compartment, from the immune cell compartment. So the complexity of the biology had to increase basically in all directions. And again, what was necessary also, which we underestimated initially when we started doing this work, what is necessary is developing all the analysis tools, which consists of developing new codes, new ways of treating data, and new ways of exploring what the data mean. And so all of this had to be developed at the same time. So the total amount of work is much larger than what I expected initially. And each level of complexity can be used to answer specific questions. And this is really the positive part of this presentation. Okay, so just to finish, this is work of many, many years with many, many people. I couldn't cite them all, but the work on the anchors to trap droplets was done mostly by this group of talented students and postdocs. The bacteria work was done by them. The double traps and the initial cancer experiments was done by them. And then the immunotherapy work was done by them. And I have long-term collaborators that have been working with me for a long time, Bastien, Gabriel Amselem, and Andrei Aristov. And this is done with many collaborations with hospitals, other academic institutions. And I have to disclose that there are several spin-off companies that are related to some of this work. And this is my group during the Christmas dinner in which I was not present, unfortunately. But thank you very much for your attention.